beginning of fourth half hour, Gr12, trace 1 and trace 2, 30 July, start.
Trying to distract you while you're talking.
Just do it on.
What?
Nah.
I'm listening.
Michelle asked me to she asked me to ask you for our test.
Okay, Mr. Florenzo will give you a test. Your test.
We can do the test now if you want.
Yes.
How do you want to?
I don't mind doing it now because they've all been emailed. And then I also made a video last night. And it was quite late and I ended up yawning halfway through. But yeah, I've sent it out to
Do you need to
everybody
get into
with
something
my video of me going over the email for, and you guys can go and watch it as well from your classroom. But we can do that now if you want. You want that now? And then in the next lesson we can do some other little bits and pieces and tie up.
Get into something now. Yeah. Do you need to get? Sorry, excuse me, yawning. Do you need to get into something here?
I think if we get into the, it says on my Dropbox oh, but hang on Google Classroom this morning is the model attached to the test. So we can go here. Okay, let me get my camera.
She did ask me for that.
They tried for 1 week and then. Where do you say it's for 1 week?
can do that now.
Are these things
Previously,
for
they were
And
you know, it's going to prepare for I think it's too short. I think it's they were trying to put me in the and it's like, you know that whole thing with that hair on the face and down and whatever, and the earrings and and you need something, trying to find something to complain about.
Amy, Randy,
Someone went to
Emily, and that's all.
That's all.
and you were like,
Okay,
yeah.
this food.
I got told that, because you know, so you have your hair down.
So,
So I had my hair down and I had it out of my face. Like I had
John, not here.
this friend of mine, she's like, no,
I have called Trisha's here.
I don't need to. I just want my earrings.
Who's in here?
Yeah,
William's here.
I don't think the issue is earrings. It's not like I want to have that many things that are
Wanda.
Yeah.
That's not bad.
Julie's not here. Here, not yet.
I have not seen Julie.
I'm not here.
No, Julia was here the other day.
Yeah.
Who was that?
Josh isn't here today. Matthew's not here. Tina.
I haven't seen Sasha.
Okay. Right.
Yeah,
Just
I
my
haven't seen
fault.
her. Sasha, Tina, take them off.
Okay, after home.
She was here.
Trisha.
What's wrong with me?
Michelle. Emily. Amy. Just see that new Ruth with yours, please. Oh, listen. Yes, I called out your name. How was it? Is it not stuck to the back of someone?
Did you make the test, Ruth?
I just ma'am.
Well done. I like the sign of rebellion at this stage.
I called out your name. So you can say it. Okay, great.
Can we read corrections on the paper?
Yes, you can. But remember, we've got to keep the paper. So that's going to go into your portfolio file. And I need to now, based on this test, Because I've already put all your marks into a portfolio spreadsheet, but I still need to go and check it. So based on this test, if you did better in this test than the other two, this becomes part of your portfolio. But it's still, this one still counts for term 2. All right, so we've already put everything in, your research assignments. Okay, but you know what, I can explain that a bit later. All right, so I'm going to, and I also want to, if I can on here, it's different.
I've got on the other one.
Oh.
Question.
I'm going to download. Oh, I see. Can I download it?
I want to download a moment ago. Wasn't offering me the option.
Yeah, I want it as a Word. Yeah, I sometimes struggle getting these things open.
I think it's a PDF.
Oh, that's why. Okay, so I can't do a split screen here, basically.
No, but I do have the.
Oh, lovely. Okay, thank you. Thank you. All right, guys. So what I said in the video, you know, this was a test on lots of stuff. Okay.
Yes, ma'am.
To me, yes, it was. You know, but everything ties together. And if you remember, you might not remember, but in February, and that test is also on, I'm sure it's on Gr12 or wherever now. I'm sure I put it on there. Yes, I have in Moodle. We did ask DNA and protein synthesis and all of that in February, but now I've picked up on it again. There are so many different ways you can ask DNA, protein synthesis, DNA replication, DNA structure. So you've done one test in February and you had some of it here. That is not enough to revise from. You really need to go and look at past papers, and you must go and look at Moodle. And I've opened a lot already, but I'm going to start opening more. And please, I said on the video, I freak out every year. When we've done prelims, we come back and I say, well, who looks at past papers? And a few little people put their hands up, and I freak out. You cannot study for prelims, am I right, ma'am? You can't study for prelims without looking at past papers, because you're not getting an idea of the range of questions that can be asked on the same topic, the different ways, and you're not getting an idea of the, you know, a lot of the questions in this test, because it was sort of like an entry-level test. You'd never written on meiosis before, you'd never written on genetics, genetic engineering, all of that. So I mean, I've got loads of different questions that I could put in here if I had 1,000 marks, and you know, from very easy to medium to whatever. So please don't think that this is going to be the only way you're going to be asked. And that's my main message, is you can't just leave it here and say, oh, no, I understand. No. You have to go look at past papers. All right, so now I need a question paper in front of me, and I think it's in my car. You don't have one, hey? From moderation the other day.
Ma'am, is it on classroom?
Everything's there. I just want to be able to read it myself. Because normally I open split screen, but I'm unable to do. Oh, you also don't have one now. Can we open the split screen? It's bugging me a bit. You can. I can just flip between the two. Okay, I just want to be able to look at this as well. All right, so okay, let's just make this a bit bigger. All right, so here, I think this was fine. The only one that I noted was a little bit of an issue was the one at the bottom. The laboratory process used to create a viable embryo, and I see there's a spelling, a grammar error there, which I didn't pick up. Okay, should just be using a body cell, but it still made sense, and an enucleated egg cell. So that process is somatic cell nuclear transfer. That's how Dolly the clone was produced. But the process is called SCNT. Did you recognize that? Okay, so put that somewhere in there. So that's where people gave me F, not E. All right, so let's go back to the model. So there we go. So it's HACGE. All right, that's the only one that I had that I noticed issues with. You can quickly squiz if you want to ask a question. All right, you can go look at this on Moodle as well. And you obviously your test is going to email to you. Okay, then the multiple choice questions. All right, let's just see there. A lot of people got all of them right. Okay, I had very high marks, or you know, where there was some confusion. All right, over here, and you know what? It's actually a good idea to go watch the video, because I've gone through every single multiple choice. Do you want me to go through everyone now? But maybe at home you can go and watch it. It's a 40-minute, sorry. It's a 40-minute video. Okay, so this was D, 1.2.1. Variation in individuals is the arrangement of those base pairs, A, T, G, C, C, G, C, whatever. Okay, this one, I put this in because we haven't had a question like that yet where you have to look at some kind of information and realize what joins to what by looking at percentages. Okay, so messenger RNA is copied from, remember the DNA molecule opens up and is controlled as to which side actually gets copied into messenger RNA. So this mRNA, if this is adenine, mRNA, adenine will bond to what on DNA? Thymine. So we've got to go to thymine, must be there. Alright? DNA, and if you look further along, uracil in mRNA would bond to what on DNA? Yeah. Okay, it's just a nice question because they sometimes ask that, and you might see different multiple choice questions, not with tables and that, but simpler questions. So have a look for those in the future. All right, then this was an easy one, and I know it was easy because we've talked about if two heterozygotes are crossed, there's a 25% chance that you end up with a recessive individual. So it was saying, what was it again? The genotypes for the parents are, so they've got that, sorry, I'm not talking nicely. So there's an albino child, there's mom and dad. In order to have a child that is an albino, mom and dad must both be heterozygous. So you might remember that summary where we, number 1, number 2, number 3, up to 6, where we did all the crosses, this with that and this with that. It was a summary. You need to be familiar with that. That heterozygous individuals, there's a 25% chance that you can get a child that's an albino. They just happen to have 4 children and 3 of them weren't and 1 was. Doesn't mean they're going to have 4 children only and 1 has albinism and 3 of them don't. Okay, so that's the next question. All right? There was a method in my madness here. So the chance of this couple's 5th child being an albino, it's still the same. It's still 25%. So if, when you do the Punnett square or the little crosses, there's 4 blocks. There's a 1 in 4 chance of them having an albino child again. You don't know what it's going to be with each conception. Okay? This one I like. Unfortunately, I wasn't able to find the sources, and I know I'm getting much better at putting all the sources in now. I have used it previously. So here's a normal sequence of DNA, and then the different sequences. Did all of you get this one right to figure out what type of point mutation it was? So that's why I put that one in, because we needed something on mutations. And I think the answer was C. Hey, you have to look at the order of the bases. I think it was C. Yes? Oh, maybe I should show you. Okay, so what question number was that? Okay, so yeah, so it is, it's A. All right, now some of them have got double ticks because they require a little bit more time. And I did go and check all of them because on one of the ones that man moderated, I made a mistake. I think it was yours, or who was it? Was it either Amy or somebody? That was the only mistake I made. So I went and checked, and that's why I put little purple ticks there just to check that I did this properly. Okay, so that's out of 11. All right, you want to just check your answers? You happy? Okay. All right, let's just go back. Question paper. All right, there, I put another one in. You all got that right? Everyone happy with that? And I've explained it. If you go look at the video, I've said, look what's matching, look what isn't, da da da. Which sire is the dad of the daughter, okay? And I put this one in. I've got quite a few variants of this. And it's a little bit tricky because it wasn't what we normally used to, which is trisomy 21. There was a trisomy 15, which I think is Patau, what's it called? No, not Jacobs. There's another name. Okay, but it doesn't matter. The only one that fitted here was an abnormal number of chromosomes. You just read through and I say this all the time, but when I get your papers back and nothing's written on your paper, I haven't got mine here, it's actually in the car. I thought I had it with me. So I go, sex chromosomes indicates female. No, cross. Chromosomes 1 to 22 are gonosomes. No, they're going to determine your gonads. So these are autosomes. These ones here. It's from a male with Down syndrome? No. An abnormal number of chromosomes is present? Yes. You should be writing all over your papers. engaging with it. Okay, we're all happy with the bacterial plasmid? Now, as I said in the video, there's lots of questions. This is a really basic level. I only had 50 marks to play with. We need to go and look at past papers where they give a whole scenario of how this transgenic organism is produced using DNA, recombinant DNA technology. There's long questions, like 15 marks or whatever. Please, you've got to have a look at more of those. Was everyone happy with this one? Okay. What was the answer? Yeah, it's that little one there with the little sticky overhanging ends, the unpaired nucleotide bases. Right, so please remember to go look at ones like that. And then this one, Duchenne muscular dystrophy. It doesn't matter what it is. It could be a disorder that you've never heard of. It doesn't matter. If you look at some of the ones that are open on Moodle, you'll find there's one about goats and things that I've used in the past. And go and look at IB ones as well. They give different scenarios. Okay? All right, so here we had some issues, and this is why I put these questions in. You've got to give evidence from the diagram that muscular dystrophy is sex-linked. So if I look there, yes, there's only males with the disorder. So make sure you just check the key first. Are the coloured in ones the ones with the disorder? I can't remember the one year they had it the other way around and confused everybody. So normal was coloured in. Do you remember that one? It's quite a number of years ago. Can't remember when. So always check the key first. There are only males here, but that doesn't mean that females could have it. We don't know that yet. That's just the little pedigree diagram that we've got now. But with sex linkage, it's more common that males have the disorder. And in this case, only males have it. All right, so let's go have a look. So I said it occurs only in males, but I put in brackets it's more common in males. Just like hemophilia in Queen Victoria's family, the females were carriers, but they passed on hemophilia to their sons. But there wasn't a hemophiliac female, doesn't mean that that's not possible. Okay, so occurs only in males in this pedigree. Could occur in females. It's more common in males in this pedigree. I only gave 1 mark, and I know I could have had more here. All right? So you will find other questions like this that you need to go and look at, and they might be out of more marks. And then you've got to explain yourself well. Okay, then the second question was, how do we know that it's caused by a recessive allele? So I actually gave evidence. I actually added this into my model afterwards. You've got to use, so what is the pedigree telling you? Okay? So yes, it's easy to say that parents who don't have the disorder pass it on to their children, but you could actually use evidence here that individual number 1, the male, okay, but hang on, we're talking about sex linkage here as well. But there are parents, like number 1 and 2, who don't have the disorder, but they are able to have children, like number 5, who does. Okay, but then we know that males can't be carriers and all of that story, okay? Because they've got one X and a Y, which doesn't have the gene. All right, so let's just look at the answer there. So yeah, I said dad number 1 normal, mother number 2 carrier, but son number 5 has it. Or a mother without the disorder can have a son with the disorder because she passes on the X chromosome to her son, but he gets Y from dad. That's what makes him male. Okay. All right. Then 1.3.2, and look at the symbols. All right, you've got to use those symbols. Some people forgot that we need to have 2 sex chromosomes. Okay, let me make this a bit bigger. I can see you squinting, sorry. Ooh. Oh my gosh. The mouse is really... Okay, so X little ny is individual number 5. Okay, let's go back. X there, the male who has muscular dystrophy is X superscript N, Y. Some people left that out. They forget the Y. You've got to have 2 sex chromosomes. And then individual number 8. Now number 8 is a female, but she has a son with muscular dystrophy. So she has to be a carrier. So only females can be carriers when it's sex linked. because the males only have one X chromosome. They either have the normal gene or they have the mutated gene. You remember that? Yes? No? All right, so then I said, let's go back to the question. The bottom here, a blood test shows that person 14 is a carrier and person 15 recently married 14, but they have yet had no children. What is the probability that their first child will be a male who develops muscular dystrophy? Okay, so you need to keep that in mind. Let's go look. So 14 is a female, all right, who is a, what did it say here? That she's a carrier and she marries a male who doesn't have muscular dystrophy. So I made it pretty easy, okay? So she is a carrier, so it's X big N, X little n, and he is, that cross is not a chromosome, sex chromosome. He is normal, so X big N, Y. Now, I do the Punnett square with my pupils, all right? But you can do that little cross, what do you call it, ma'am? I've forgotten. When they do that, I call it foil, genetic foil.
That's what I normally refer to as genetic diagram, although then I know what Punnett square is. And then I just say the crissy-crossy one.
Okay, all right. Okay, so who does the crissy-crossy one? And you're allowed to do it, but when I was marking, I think it was about 3 years ago, when you do, I'm not going to write this whole thing, so X, I don't want to do the whole thing. All right, X, Y, X, Y. You've got to show, and underneath you've got to write the answer. What about that? There, underneath you've got to write the answer. And there, and there. Because when we marked, and people had written that answer here, or this answer here, they didn't give marks for it, because they say you're being sloppy and it's not linking up nicely. Can you see what I'm saying? Just make sure it nicely matches and everything. Okay, so they don't mind if you do it. I just do the Punnett square, but you can do both. All right, so this is what we get. So sometimes they give 1 mark, sometimes 2 marks for the parents. It depends. Okay, please try and remember to write the parents first and not only in the Punnett square or only at the top. All right, and then you get a mark for working out the genotypes of the offspring. Now, I did not give a mark if you just, like any normal problem, got the answer and said there's a 25% chance of having a male with ND. We need to see that you've linked it to there. They were quite adamant about that. Okay? So if you've just written that and written the standard issue answer that there's a 25% chance of having a male with MD, but I don't know that it matches that or that or that or that, they didn't give the mark. So I'm just warning you. That's the one paper that I was marking the one time. Yes?
No, that's really underlined or highlighted. So
We accepted anything. It was highlighting, it was an arrow, or it was a summary at the end. Therefore, males with the genotype X superscript little n, Y, there's a 25% chance of having, an offspring with muscular dystrophy. But they didn't give you a mark if you just standard issue answer. Okay.
Now, how are we meant to know that we're meant to link it? Because when we're doing problems,
Because we've been saying it.
But we didn't know that.
Guess what? You now know, and guess what? It's going to be 0.0000 something of this term, but in your prelim it's going to count lots more, or your final. Okay.
Sorry, will you monitor us?
Why you have to physically link it to that? Because when we did it in class, we just looked at the Punnett square and then wrote it out. We didn't highlight it or draw an arrow.
But you had to say it in your answer somehow. So sometimes it just says do a genetic problem, and then sometimes it was more specific where it says a male, what's the chance of them having a male with this disorder? Then you've got to be more specific. Okay. It's a better lesson to learn now than in prelims, and then you're already, you set for finals. It really is. Okay. All right. Okay, this next question, oh gosh. I was a bit worried about this, and when I was going over the model last night, I wasn't quite sure why we were getting these wrong. Do we just not like them?
no, they're not.
You have to work with these questions. Am I right, ma'am? They just go...
Your paper 2 examiner is extremely fond of them. I moderate all his class tests. He's very fond of them.
And last year's paper, there were so many of these little tables all
Yes.
over the place.
Yes.
And we were like, whoa. But you have to think. You have to get them right. You've got to write on here. You can't just like, oh, I think it's 1, or I think it's 8, or okay. So please, you've got to work with this. All right. So the answer I think was A, B, D, C. Okay. Yeah, A, B, D, C. All right. And okay, so hydrogen bonds. Remember, they link the nitrogen bases. So tick. Links cytosine to guanine. Tick. Links deoxyribose to sugar phosphate. No, that's a sugar phosphate bond. That's the sides of the DNA ladder. So it's only number 1, A. You're writing A next to the answer. But here you had an answer sheet because of lockdown. You will get a column here where you're going to write A. Okay. Then, okay, now, centromere, a point at which 2 homologous chromosomes. Homologous chromosomes, you know, there's 2 pairs. The only time they ever link, you know, for a short time is crossing over. They're not linked. But point at which 2 identical chromatids are joined is a centromere. But how many chromosomes do I have in my arms? 2 chromatids, 2 DNA molecules joined by a centromere. How many chromosomes? 1. Thank you. Okay, so you count the number of chromosomes by counting the number of centromeres. Okay, so centromere, the point at which 2 chromatids, DNA molecules are joined. Because remember, it was 1 DNA molecule, opened up, got copied. I should just draw this, I suppose. Okay, 1 DNA molecule coils up, but it also gets copied, so you end up with 2 of them joined by a centromere. 1 chromatin thread, 1 chromosome, still 1 chromosome, but it's a replicated chromosome. Because the centromere then divides, and the chromatids, the daughter chromosomes, are pulled into daughter cells. Okay. So the answer there is 2. So this is where people get confused. So it's cross, not joining homologous chromosomes. That would only be chiasmata, crossing over. 2, yes. B. Okay. The next one, oh boy, this is why I put this one in. DNA replication only occurs at either the beginning of mitosis or meiosis, right at the beginning. Okay? It does not occur before meiosis 2. Only at the beginning of meiosis, and it doesn't occur before protein synthesis, because that's gene expression. mRNA copied, transfer RNA, amino acids. Yes?
You see, I think maybe, well, I personally got confused with the English. Because if you say before meiosis 1 and 2, it's before both. That's how.
That's what I mean.
Yes.
Yes.
That's how my
And it doesn't.
when
It only occurs at the beginning of meiosis 1, where the chromosomes double up. So they are now.
Does it not occur before?
Okay, you know what? We'll look at the meiosis picture just now. So it only occurs, DNA replication, at the beginning of the whole of meiosis, and at the beginning of the whole of mitosis. So yeah, it was saying that it occurs before meiosis 1 and again before meiosis 2. And it doesn't occur before protein synthesis. So neither of them are correct. D. Yes.
I think what Ruth was trying to say is she took, it's saying meiosis 1 and 2 as a whole process.
Yeah, I can see that.
That's why we got confused.
But then wouldn't I say it occurs before meiosis? Why would I say 1 and 2?
Maybe just being specific.
It could be a trick, even. It's just the English was very confusing.
Yeah, but can you see what I was trying to say there though? It occurs, maybe I should have said it occurs before meiosis 1 and before meiosis 2.
Yeah. Then we would have.
But it's weird to say it occurs before meiosis 1 and 2 because it just occurs before meiosis, doesn't it?
I don't know, some people just call meiosis like
Okay. Alright, one second. Okay. Turner syndrome. I kind of went last night when I did the model here. And then I read up on Turner syndrome for like half an hour and then I ended up going to bed late. Okay. So Turner syndrome, remember that is where they have X and nothing. But there are lots of variations, alright? So, someone with Turner syndrome, will their somatic cells still be diploid? Do they still have 2 sets of chromosomes? Do they still have chromosomes from mom and chromosomes from dad in all their cells? Yes. But do they have an abnormal number of chromosomes per cell?